In this video, we're going to look at nucleic acid hybridization and denaturation. When two nucleic acid strands are joined in a double helix, they are said to be hybridized or annealed. When DNA in a cell is not being used for replication or transcription, it is fully hybridized, as seen here. However, when the cell needs to undergo transcription or replication, it will begin to separate the strands. This is called denaturation, or in some contexts, melting. Denaturation can occur due to a few different factors like enzymes, high temperatures, pH fluctuations outside normal physiological levels, and salt concentrations. Now, let's focus for a second on temperature. And more specifically, let's discuss melting temperature, which is the temperature required to separate half of the double helices in a sample into single strands. There's a lot going on in this image, so let's break it down. As we can see from this example, DNA remains fully hybridized until a specific point, and then the DNA rapidly melts over a small temperature range until all of it is denatured. This point, where half of the DNA is denatured, is defined as the melting temperature for that particular DNA. Increases in the number of hydrogen bonds between bases increases the stability of the double helix, which means melting temperature will increase if more G to C pairs exist. One reason for this is that binding between C and G involves 3 hydrogen bonds, whereas bonding between A and T, or A and U, only involves 2 hydrogen bonds. Another way to increase the melting temperature is to use longer double helices, since the sheer number of base pairs, and therefore the total number of hydrogen bonds, is higher. When denatured DNA in a solution returns to normal temperatures below its melting temperature, it will begin to rehybridize or reanneal. When it comes to the reannealing speed, there are a few influencing factors. The length of DNA can be an influence. For example, longer strands take longer to anneal because there are just more bases that need to come together. pH also influences annealing. When the solution pH is near 7.4, annealing proceeds more readily. Lastly, salt concentrations play a role in this process as well because DNA double helices are more stable when the positive ions in salt can interact with the negatively charged phosphate groups in DNA. These factors must be considered when creating optimal conditions for molecular techniques that rely on denaturing and hybridizing DNA. For example, PCR, a technique used to amplify the amount of DNA in a solution, begins by denaturing a DNA strand. This is done by heating. Once denatured, the individual strands are annealed with short nucleic acids called primers. To allow this annealing, the solution is slightly cooled. These primers allow elongation to occur when proteins called DNA polymerases create complementary strands to the denatured DNA. So now we have 2 DNA molecules instead of 1, and this process can be repeated to amplify the amount of DNA in a solution. The denaturing and annealing are controlled by alternating between higher and lower temperatures. So an understanding of how nucleic acids melt and anneal is extremely helpful for techniques like PCR, which can be used to diagnose many different genetic disorders. Keep studying!